All right, a little loud there. Is that better?
No, still pretty loud.
Still too loud. Can you modulate this before I kill people up in class here?
Can you see her at the back?
How's that? Still too loud or good in the back? Okay. I will try not to raise my voice, which is unfortunately a normal mode for me. So yeah, she's going to turn me down. Welcome to BIS2C. Hope you're in the right place. I'm Jonathan Eisen. I'm a professor in the evolutionary ecology department and in the medical microbiology and immunology department, and I'm going to be teaching for part of BIS2C, and Professor Susan Keene, who we will hear from in a minute, is going to be teaching the other part, and I'll tell you when we go through who's doing which parts of the class. So what I'm going to do for every lecture, and what I'm going to just try and do to get you, into the mood in the first couple of minutes of the class here, is remind you what we did in the previous lecture. Previous lecture should have been BIS2B for everyone here. If you have not completed the prereqs for this course, you probably shouldn't be here, or unless you have gotten some sort of exception for other courses that you've taken, but you need to have taken the other courses in the series. What we're going to do today is talk about the administrative details for the class, give you an introduction to some of the different things that are going on in the class, the big picture of the class, and then for the latter part of today, I'm going to talk a little bit about phylogeny and phylogenetic trees. And then in the next lecture on Monday, we're going to talk in more detail about phylogenetic trees. I'm also going to try every lecture to remind you what the reading should be. So for the first 4 or 5 lectures, the reading that you need to complete, hopefully before Monday's class, is chapter 22 in the textbook. And it would be best if you read that before the lectures on Monday. And then in the course description on the Smart site, there's the other chapters that you need to read. And generally, when we switch topics, like we'll do a few lectures on phylogeny and phylogenetic trees, and that's chapter 22. And then we're going to switch to talking about microorganisms, and that's going to be a different chapter. And it would be best if you tried to read those before the lectures. And you're going to hear a little more about how we're going to incentivize you to read some of those through quizzes and through some online activities. So I'm also going to try and give you an outline every time. So again, we're going to go through some administrative details, and then I'm going to talk in more detail about phylogenetic trees for today. So what I want to do first is just give you the big picture. What is the point of this class? And so the main theme you will see throughout this class relates to biodiversity, to the incredible diversity among organisms, between organisms on the planet. And that diversity is seen at many different levels. You can see diverse behaviors, morphologies, biochemistry, molecular biology, all sorts of different form and function diversity across the tree of life. And so here you have representative of a group of organisms we'll talk about in about a week, the archaea. This is an example of a bacterium, and this is an example of a microbial eukaryote, an organism with a nucleus, but it's small and single-celled. And that's what I focus on in my research, is microorganisms. And I'll be talking, teaching in this course a lot about the microbial parts of the tree of life. Of course, a lot of what you're more familiar with is going to be the multicellular organisms on the planet, the plants and fungi and animals, and the diversity of those, which is also extraordinary. And we're going to spend some time on each of these groups of organisms and go through their diversity and again, examples of the diversity of their life cycles, diversity of their physiology, diversity of their behavior, diversity of their any type of process that you could possibly imagine. And this diversity is completely overwhelming. There are estimates right now that there are tens to hundreds of millions of species of organisms on the planet. And they're all different from each other. And this diversity is just sort of astonishing and overwhelming. And what we're going to do in this course is organize this diversity with phylogeny. And so phylogeny is, and I'll define it again in a few minutes, is a representation of the history of organisms. And this phylogenetic tree, which I've taken from chapter 1 in the book, is a representation of the evolutionary relationships among all organisms on the planet. And that, when we include all organisms, that's called the tree of life. And that's where the name for this course comes from, Biodiversity and the Tree of Life. We're going to talk about biodiversity and learn about biodiversity, and the organizing theme behind this is going to be the evolutionary relationships among organisms. And what we're going to do is basically interweave and alternate between two major categories of things you're going to have to learn. And the first is a little bit big picture, which is concepts about biodiversity and phylogeny and the tree of life. And so in terms of phylogeny, we're going to learn about phylogenetic trees. What are they? What do they mean? What are the different components of a phylogenetic tree, and how can we interpret them? We're going to learn about how you can use phylogenetic trees to characterize and study the diversity of life. And we're going to learn about how, if you have data from organisms, either modern organisms or fossil data from past organisms, how we can figure out or infer what their evolutionary history is. And so that's called inferring phylogeny. And we're also going to occasionally go back and forth talking about what we call tree thinking, which is really the different uses of phylogenetic trees in the context of understanding biodiversity. We're also going to learn facts about particular organisms. So not just this big picture conceptual stuff, but we're also going to have to learn certain facts about organisms. We're going to learn about the major lineages of organisms on the planet as we currently understand them. We're going to learn about key features of these organisms. So what distinguishes angiosperm from a gymnosperm? What distinguishes a bacteria from an archaea? What distinguishes different types of organisms from each other? What are the telltale features? What are the interesting features? We hope we will find many of them interesting. And we're also going to talk about major events in evolutionary history, transitions between particular types of organisms. So again, you just have to sort of realize that there will be some of these key facts that you're going to have to understand, and also the big picture concepts. And we're going to try and take you through, when we go through a lot of detail, we might show you a picture with 17 things labeled on it, and we probably don't expect you to know all 17 things on that slide. We might tell you which things are important and which things are not important. We will try to give you guidance to the facts that you will have to know, as well as the concepts throughout the class. So that's the big picture of the course. And now what I want to do is take a step back and talk about just administrative details, because there's lots of stuff we have to get going for all of you and make sure it all works in time for basically next week and the labs and other aspects of the course. So again, I'm Jonathan Eisen, professor in various parts of this university. And this is Susan Keene, who's going to be lecturing a little bit, and she'll tell you about some other things in a few minutes related to the course. Joel Ledford is the course coordinator, the real nuts and bolts behind the course, and for handling all sorts of details with running the labs and grading and coordinating everything associated with the course. And you'll hear more from him in a minute. Corinne is here. Oh, there, a different spot than earlier today. Is the course assistant. Her main role from your point of view for now is going to be related to registration details. So if you're having problems with registration or other things associated with registration, you need to get in touch with her. That's going to be her responsibility for the beginning of the course. Giuseppe Vizzoli is over in the corner. He's the head teaching assistant. So he's the boss of all of the teaching assistants. And you will meet your teaching assistants in lab next week, and a few of them are here, and we'll introduce them in a little bit today. So I get many questions about this every time the course gets listed in the course catalog. The course meets 3 times a week at this hour, Monday, Wednesday, Friday. And there's also listed in the course catalog this 4th discussion section. It's not really a different type of class than the rest of the class. It's going to be basically like this, another mandatory lecture for the course. We will try and not have it be too painful when it's Monday at 6:10 p.m., but there still will be regular activities associated with the course, things you need to know, just like every other lecture. So I don't know why they have to call this a discussion section in the catalog, but it's not really separate from the rest of the course. Midterm number 1 for the course is going to be in the second of the Monday classes for you guys on October 22nd. Midterm number 2 is going to be in the regular class on Wednesday, November 14th, and the final exam is going to be December 13th, 10:30 to 12:30. If you have any conflicts with any of these dates, you need to talk to us now. If you have officially sanctioned conflicts like some sort of school activity, we may be able to grant a makeup exam, or we may just say you're going to get all the points from the other tests in the class. If you have tickets for a vacation, that's not going to be granted a makeup for the exam. So you need to talk to us immediately if you have any conflicts with this, and we need to set the wheels in motion for figuring out what we're going to do with those conflicts. So I assume most of you are reasonably familiar with SmartSite. We are going to use SmartSite for many things associated with this class. We are going to be posting lecture slides there. The audio for lecture will be recorded and posted as a podcast to SmartSite. The chat room, we will try our best to answer questions in the chat room if you post them. Sometimes they get lost in the thread, but we will try our best every once in a while to answer questions that are there. You will do your pre-lab activities via SmartSite, so you have to become familiar with SmartSite. We're going to use it for lots of things associated with this class. The textbook is the 9th edition of this Sadava Life, the Science of Biology textbook. It's really important to have the 9th edition. All of the reading, all of the requirements associated with the course, many of the activities for which you will be getting bonus points and other things for the course are geared around the 9th edition. If you have the 8th edition, we are not going to try and figure out what is different between the 8th edition and the 9th edition for you. So you really should try and get the 9th edition. There are copies in some of the libraries on campus if you don't want to buy the 9th edition for whatever reason, but I really recommend that you try and get the 9th edition of this textbook. Assigned reading you'll be told about in lecture, and it's also on the syllabus. And occasionally you might get an additional reading like a newspaper story or something to that effect. And I'm going to turn it over now to our course coordinator here, who's going to give you a little more detail on the lab and some other parts of the course.
So as Professor Eisen said, I'm the course coordinator, so I'm essentially responsible for organizing and maintaining the labs and supervising the TAs. Our job is to provide you with a good, interesting experience so you can practice some of the things that you learn in lecture, and you're going to get lots of hand-on activity-based experience in class. For enrollment, you're going to see Corinne once again. When you first come to class, if you're on the wait list, what I'd like you to do is please wait in the hall. Do not go and sit inside a classroom unless you are officially enrolled. Right? Wait outside for Corinne.
For the laboratory.
Sorry, yeah, for the lab. TAs, we have a great group of TAs. This quarter, I'm going to introduce some of them to you if they're here. We have Melody here. Not. Nafisa. Giuseppe, you already met. Sang is here. Rocky? Nope. Aaron? Nope. Mijen is here. Great. Elizabeth? No. Tim is here. Jamie? Nope. Kevin? Emiya is here. Alex was here earlier, but I don't think he's here today, or now. Daniel, great. Mark, Marasano, I saw him somewhere. There he is. And Srija. Excellent. So for labs, a couple of things. First, make sure you pick up a copy of the lab manual as soon as possible. Raise your hand if you've already done so. Outstanding. Keep in mind, your first pre-lab is due by Monday at 9 a.m. I put the link up well in advance, so if you haven't started, you have a full weekend. The first lab is a little long, but it's fundamental to the class. Okay, so make sure you get it done by 9 a.m. At 9:01, I'm taking the link away. It's the class policy that if you do not do the pre-labs, you're paying attention, I hope, if you do not do the pre-labs, you do not get points for the lab, right? You are free to go to open lab, but you will not get points for the lab. Make sure they're fundamental to your understanding of the material that's presented to you in your lab sections. There have been some stories in the past about, I can see the time you submit, right? You can't start this at 8:55 and finish and submit by 8:59, right? Make sure you get it done well in advance. Sometimes things get lost in the internet ether. That happens. It's not common, but it does. If you're worried, what you should do is print out the copy of the receipt that you get when you finish the pre-lab, or you can take a screenshot just in case it gets lost. Okay? There have been some browser issues. In the first pre-lab, there is an animation, and for whatever reason, it's not working well with Google Chrome. It's a Chrome issue. Smart Site accepts no responsibility. Neither do the other people that are sort of in charge of this thing. So I have tested it on Explorer and Firefox and Safari, both on Macs and PCs, and everything else seems to work fine. So just leave the Chrome alone. Prerequisites. If there were problems with your prerequisites, you should already have been contacted by Hurin. But basically, you need a C- in 2B or better in order to go to the class. If you don't have that, Hurin will probably be contacting you if she hasn't done so already. Class communication. We use Smart Site regularly, so please make sure you go and check. Raise your hand if you've already checked. Excellent. I've also made a Facebook page for the class. The link is posted up there, so if you're a Facebook junkie, you check your Facebook more than email. It's there for you. And I'm going to put short little announcements up on that page. Instructor office hours, my office hours, and TA office hours are already posted on the Smart Site as well. It's a PDF. Download it and have a look. One thing to, again, keep in mind is that we have open labs on Friday from 9 to noon. So if you do miss a lab, you won't get credit for it, but you're still responsible for understanding the material. So please come to open lab. TAs. The TAs have, I have high expectations for the TAs, and they have high expectations for you. So they should be getting back, returning your homework back within a week, and they're there to provide you and facilitate your learning, but they need your help, right? Your help means go there, be an active, engaged learner. Create a good learning community in there. It will make a big difference in this class. If you feel like you're not getting homeworks back or something is going wrong, please feel free to contact me. Last but not least is my soapbox spiel. So how many people are interested in being scientists or being involved in the scientific profession directly, either medical school or otherwise? Excellent. Excellent. Yeah, so what I would say to you, what I'd urge to you is that you have a duty to be truthful and honest, especially in your science itself and your data itself, but also in all the things that you do. So, my main voice here, do not cheat in here. Right? If you cheat in this class, I will refer you to SJA. That includes pre-labs, homeworks, and clickers. So if your buddy is sick and he says, hey, will you do my clicker for me today in class? And we see you with 2 or 3 clickers, and it has happened, we're going to refer both of you to SJA. Right? So please, be honest, and I think it'll carry you a long way. Are there any questions that I can answer? Yes?
So do we need to access BioPortal?
We'll talk about that in a minute. Yeah.
Is my slide there, buddy?
Sorry. It is there, it's just not showing.
Okay. This is the Smart Site. I just wanted to point out the pre-lab is there. Again, the screenshot thing is really helpful if you are at all worried about protecting yourself from the missing the lab. Okay. Any other questions? Yep.
Do you have to go to the discussion section that you're assigned to, or can you go to any of them?
You need to go to the discussion section you're assigned to.
Yeah.
Right? You need to go to the discussion section you're assigned to.
Or you won't get any points.
Or you won't get any points. Yeah.
So do our clickers need to be registered by Monday?
I know people have asked me about questions
Yeah.
about online registration for clickers.
So here. You need to get clickers by Monday. We're going to try in the first section on Monday to practice using them and go through to make sure everybody has them working in the first section on Monday. So we will discuss, all the things you need to do for them in the class on Monday, if that's okay. So you don't need them today, and we're not going to count them for points in the first section on Monday. But in the second section on Monday, we might use them for a couple of clicker questions. So you have to have it by the morning session, by this session on Monday, so that we can make sure they're all working for everybody. And I'll tell you a little bit more about clicker points in a second, but Professor Keene, I wanted her to talk about the bioportal for a second here.
Okay, great, thanks. Okay, so bioportal is the resource that you get when you got the textbook. And with any luck, you all got the textbook when you joined BIS2A, and you got the textbook version that we made for you that was fairly cheap, like about $100. And that included 18 months of bioportal access. But you need this little card thingy that was inside the book. All right, does everyone remember the little card thingy? How many people have already registered their little thing? Oh, okay, so 2/3. So if you haven't, you need to search your dresser drawer or wherever it is, look in the closet and find the little card thingy with the access number, yeah?
So if we registered for a previous class, we don't need to register again?
Right, if you registered for a previous class, you don't need to register for it again. And I'm pretty sure, and I'm not positive, but I'm pretty sure that if we assigned a chapter that 2B also assigned for mastery, which I'm getting to, I think it just transfers over. But anyway, so what's on bioportal? Bioportal includes animations that you can look at about the material, some of which we'll show in class, little videos of the things, and 2 kinds of quizzing. Self-diagnostic quizzes, so after you look at the chapter, did I get most of the major points, you can take a diagnostic quiz. We're not giving you any points for those, but you can just do them to see if you get it. What we will do is give you bonus points. So the total number of bonus points this quarter is going to be 25, okay, for 2 things. Clicker questions and bioportal, okay? And on the bioportal, it's the PrepU. And PrepU is an adaptive, how many people have used PrepU before? Maybe a quarter or so. Okay, PrepU is an adaptive quizzing system that goes with the chapters. And basically what it is when you decide to do, you read the chapter and then you decide to take the PrepU quiz for that chapter. If you, the way it works, what's adaptive about it is that the computer decides, based on the first quiz you take, how much more quizzing you need to show that you know anything, okay? So you go in and you decide, my first quiz should be 10 questions, or my first quiz should be 20 questions. Say you do 20 questions, and you get 18 of them right. Then it might make you do another 2 quizzes and say, okay, this person's getting a lot of them right, they can reach mastery level 3 pretty quickly. If, on the other hand, you did your first quiz and only got 2 questions right, the computer would decide this person really doesn't know very much, they're going to need to be quizzed quite seriously. And they might make you do 60 or 80 or 100 questions to get to mastery level 3. Okay? So the approach where you don't bother to read the chapter and just wing it causes you to have to do zillions of questions, which is not a good way to get to mastery level 3. Okay? So mastery level goes all the way up, I think, to 9. Okay? We've only set it to 3. It's pretty basic. I think in 2A this quarter they moved it. Used to set it to 3. They moved it up to 5. But anyway, we're at 3. So you go in, you do your quizzes, you reach mastery level 3. There's a tab there on your own bioportal. You go in, you enter your ID, then there's a tab for you that tells you which ones it's recording that you completed. Okay? We're dividing them into 3 sets. The first 4 chapters that are due before midterm 1, and on, I think it's 9 a.m. on the morning of the first midterm, you cannot access those chapters anymore. So if you did them before then, you can have 4 points. If you didn't do that before then, you can't have 4 points. Then there's 4 more chapters between midterm 1 and midterm 2. They close down at 9 o'clock on the morning of midterm 2. And then there's 3 chapters before the final. So if you do each chapter that you get to mastery level 3 at, you get 1 bonus point. Okay? Anybody got questions about that? Yes?
If we're past the 18 months, we have to purchase another access code, right?
Yeah, you would. You took 18 months between 2A and 2C?
Yeah, because I couldn't get into class. I had to try to do the whole hallway thing.
Oh my goodness. Okay. If you're gonna, I'm gonna try, so the full price, so the deal is we got a really low price on the whole packet. And if you have to buy BioPortal all over again, it's $88 for standalone, because what happens is, and I try to tell people not to buy, like if you buy a used textbook in 2A, then you pay the like $50 for the used textbook, then you got to pay $88 for BioPortal. That's way more than we would have charged for the whole kit. So I will ask them if they will, since you only need it, assuming everything goes well, you only need it for 10 weeks, right? I'm going to see if I can get them to cut it in half or something just for a 10-week access. Okay, I will find out how much they're willing to cut it down and make it really tight, so you won't get mailed. And do you have proof that you bought it once?
Well, I don't even know. I know I had it, and then I registered it, so I had already registered it.
Okay. Well, if you have your reg code, you can send me that. Is anybody else in that boat? 18 months is a long time. Okay. If you can prove that you already registered it, let me know. Maybe I can make them even reactivate because you did pay once. I'll find out. Yeah.
If you buy the access card just on PrepU,
yeah.
Is that okay?
No, because the access card just on PrepU is only $18, but it doesn't connect to a grade book. I mean, sure, but it won't help you with your bonus points. Okay, so I have to try to find another way. Most people, I think, probably have their access cards somewhere. Hopefully still inside the sleeve of the book. Anybody else got a question? Yeah.
So you said bonus points could be from PrepU or clickers?
And clickers, yeah.
And clickers. So does that mean clickers are just bonus points?
Clickers have 2 uses, bonus points, so we're going to do 2 things. In class, we'll both be asking questions once in a while, and they will have bonus points, and if you get them right, you get 1 raw point, and if you get them, if you get them wrong, you get 1 raw point, and if you get them right, you get 3 raw points, and then we'll take the total number of points and add it up over the quarter, and that will be boiled down. If you got every question right, you could get 14 bonus points for the clickers. Okay, so that's optional, and the PrepU is optional. But what is not optional with the clickers is that there will be in-class quizzes, and Jonathan will talk about those in a second. Professor Ivy will talk about those in a second. Okay, but so there's 2 things, and the bonus points are optional. If you don't want to try for them, don't try for them. Okay, but they cover a little multitude of tiny sins on midterms and other places. Any questions? Yes.
So how many total bonus points are there for PrepU?
11 for PrepU and 14 for clickers, totaling to 25. But it isn't just 14 questions from clickers. It's going to be a lot of questions boiled down to 14 points. Okay? Every once in a while there's someone in the class who says, I answered 14 questions, I'm done now. And I'm like, not exactly. Okay, so anybody else? Okay, you're up.
All right, so the main point behind doing these tests for the different chapters in the book and doing the clicker questions and also doing these in-class quizzes is to try and keep everybody current, up to speed with everything that we're doing, in a way incentivize you to read the book and to follow along and be ready during class so that come the end of the period when there's a midterm or the end of the quarter, you're not going to be really far behind. So in most cases, like the bonus questions and the clicker questions, many of them will not be the most difficult things in the world. They're more, and you will get credit for participation in the clicker questions, partial credit. So we're really trying to give you rewards for staying up to date and attending class and give you this bonus for doing that. And the in-class quizzes will be sort of a similar thing. We will tell you in advance say in the next lecture, we're going to start off with a quiz about, page 324 to 326, or something to that effect, or something about something we talked about in the previous lecture. Maybe we'll do a quiz on that. So we'll give you a little heads up on the quizzes, and then hopefully you can accumulate some points through those by participation so that maybe you won't be quite as stressed come the final exam time or so on. So in terms of the total points in the class, this is a summary of how it's going to be balanced. The lab participation, attending and doing all the activities associated with the lab, is going to come up to 105 points, and then 45 points for a lab practical at the end of the quarter. The midterms are each worth 75 points. The in-class quizzes are worth 50 points total throughout the entire quarter, and then the final is 150 points. And again, on top of all that, you have the potential to get 25 true bonus points for the course. Was there a question about?
When you guys have your in-class quizzes, do you prefer to hold them like in the first 10-15 minutes of class or towards the end?
I don't have a great answer. I mean, probably prefer to hold them at the beginning, but it could vary depending on the timing. We have to coordinate two sections, so sometimes section A gets a little ahead of section B, and we try and have the quizzes sort of at the same point sometimes, so it may be hard to synchronize. I mean, my goal would be to do them at the beginning. Just technically, it's a little easier, but I don't really know. Any other questions about the points for the class? Any other questions about administrative details? Yeah?
I just noticed on Bioportal lists the instructors as Ice and Keen and Moore for this section.
Yeah.
Is there Professor Moore, or is that not?
Professor Moore is not teaching. It's just the two of us. So you're stuck with us. Brian Moore is really excellent. I taught with him last fall, but he's in desperate need of not teaching a giant introductory course for his sanity. So we're doing it together. If you came here for Professor Moore, we'll have to talk about it later and direct you to his office or something. All right, so now what we're going to do in the remaining 17 or so minutes is get into the meat of the course. So what we're going to do now is talk about sort of phylogenetic trees and a little introduction to phylogeny. So again, just reiterating what I talked about before, there's this incredible diversity of life, form and functional diversity. Much of it you're familiar with, some of it you will be unfamiliar with, and we're going to go through that diversity of life and orient it relative to phylogenetic trees of organisms. The phylogenetic trees are not, the only thing we expect you to learn about. They're going to be the scaffold that we use to understand the diversity of life. And if people have ever read The Origin of Species, if not, you probably should, but if you have ever read The Origin of Species, it's, if you flip through it and you say, where are all the pictures? There aren't any, except one. There's one drawing in the entire book, and it's an evolutionary tree that Darwin drew. I've redrawn it here with a little bit of color, but it's basically the same tree as the one on the right. And the reason Darwin used that as the only figure in the book was the key aspect of understanding the diversity of life that he really brought to the world was that the branching pattern in the evolutionary history of organisms and the divergence of organisms from common ancestors is really how you can understand and interpret the full diversity of life on the planet. And the way we do this is through, I'll just go back for 1 second. The main way we try and understand this history is through a diagram like this, what's called a phylogenetic tree. And it's a sort of simplification of the history of organisms, and we'll talk a little bit about this simplification in a minute. But I've sort of tried to summarize some of the key features on this slide. So a phylogeny is a description of the evolutionary history and the relationships among organisms. We also occasionally will use the term to refer to the evolutionary history of parts of organisms. So we can talk about the evolutionary history of genes within organisms. And sometimes they don't exactly follow the evolutionary history of the species that those genes came from. So we sometimes talk about phylogeny of genes or phylogeny of species. The phylogeny is portrayed in this tree diagram, this branching diagram that we will talk about in probably more detail than you ever imagined over the next few lectures. We will see hundreds of phylogenetic trees in this class. Sometimes we use them to represent the evolutionary history of a group of species in one genus. Sometimes we use them to represent the evolutionary history of an entire order of organisms. Sometimes we use them to represent the evolutionary history of everything. And so this tree of life concept, which we will refer to a lot, especially in the beginning of the class, is in essence a representation of the evolutionary history of everything on the planet. And this is covered in chapter 22 in the book, and for the first 4 lectures or so, that's the material in the reading that you need to read in order to understand more deeply the material that we're going to go through in the lectures. So this is an example of a phylogenetic tree, one of organisms that you may be familiar with or not. So this is a phylogenetic tree of some of the primates, and this tree diagrams a model of the history of these organisms. So scientists got together, in some dark room. So there's a model that people have drawn up of the evolutionary history of these organisms, and it's represented in this tree. And what this tree shows is a couple of key features that you should understand. For any tree that we show in the class will have these same types of features. So in many of the trees in the course, we will draw them in what's called a horizontal tree. You will see other forms of trees in the next lecture, but in this form it's called a horizontal tree, and that's because time goes from the left to the right, the past to the present, along the horizontal axis here. And that's called a horizontal tree. And if we go to as far as we can on the left of a horizontal tree, what we see is the root. This branch here is called the root of the tree, or the root branch of the tree. And this point here is called a node. Anywhere where a branch going from the left splits into 2 descendant branches, that split occurs at a node in the tree. So here's one node, here's another node, here's another node. And those splits basically represent the divergence of ancestral lineages. So in this case, there was a common ancestor of humans and chimpanzees sometime in the past, and that common ancestor split into 2 descendant lineages. One of them led to humans, one of them led to chimpanzees. In this tree, there's also a common ancestor of gorillas and humans and chimps, and there was also a split from that common ancestor in the tree. So it's important to sort of think of trees in 2 different ways. One is going from the past to the present. When you do that, the nodes represent the split of one lineage into 2 descendant lineages. If you go from the present to the past, the nodes represent the most recent common ancestor of the organisms to the right of that node. So this node represents the most recent common ancestor of humans and chimps. And here is the most recent common ancestor of humans, chimps, and gorillas. And every tree that we show will have this same basic structure. Yeah?
Will there be times where nodes will have more than 2 species splitting off into it?
Yeah, so the question is, will there be times when nodes have more than 2 lineages branching off of them? There are. They're called polytomies, and we will talk about them next week. They're mostly used for testing purposes, or in cases where we just don't know what the order of branching is among organisms. In some of the trees that you will see in the class, one of the axes will be labeled with time, and that's usually done only in cases where we have good information about the explicit time when certain events may have occurred in the past. So if we have a good fossil record for a group of organisms, for example, we may draw onto the x-axis actual time events. And you can figure out when particular events represented in the tree occurred by just drawing a line down to the x-axis in this case. So you can figure out when certain events occurred. In many cases, we will not draw time on the x-axis. It will just be a representation of evolutionary progression without a scale in some cases. And in other cases, the scale will be degree of change among organisms. But we may not have time that we can assign to those points in the tree. So we use these phylogenetic trees for many, many purposes, and you will see them again throughout the class. But you should understand when you see them that they are oversimplifications of the true history of organisms. They are incredibly useful, as you will see throughout the class. But that doesn't mean they are trying to represent everything that's going on. Just like a highway map does not represent every tiny little turn that the road goes on, although I guess if you use Google Street View, you might actually be able to see that. But imagine Google Street View for phylogenetic trees. What they're going to look like is something like this. Instead of the simple branches, what you would see with the evolution of species is more something akin to this. So let's say you have an ancestral population labeled by A, or an ancestral species. And this is a group of bunny rabbits, for example. And they're, as bunny rabbits might do, mating with each other and producing offspring. They're recombining. You have a male and a female producing offspring. And if you tried to draw this in terms of the history, you would have branches crossing over each other all the time within this population. Like when you draw sort of family tree diagrams, you can't draw them in one straight line because you have sexual reproduction and you have all sorts of other things going on in the population. And at some point, this lineage splits into two descendant lineages, and you get eventually two descendant species, B and C. So if we represented every single individual in the populations of these bunny rabbits, it would be a mess. But what we try and do is represent, in essence, the general direction of the path of these organisms, the average of what's going on. So we draw a line down the middle of this population, in essence, and it represents the core of the evolution of that lineage or of a particular species. And we don't want to confuse things by showing all of the detail, so we throw it away. But it's there, hidden behind the phylogenetic trees that we draw in most cases. And so we throw it away to get this resulting tree, in essence, and we can add the node, highlight the node on the tree, just like in the primate evolutionary tree that I showed before. And this, of course, occurs at every level. So if you had 4 descendant taxa, there would all be a lot of complicated population movement of genes and organisms, but we're not going to represent that. We're going to throw most of that away and show the core, the evolution of the species sort of as a concept, even though there's a little bit of detail that's not shown in here. And again, you can add the nodes. So if we did this for the primate tree, it would look much more complicated. And this is actually probably a simple representation of what it might look like, but it would certainly be a little more complex. And again, we just don't show that, but you should remember that that's there behind the lines when we draw evolutionary trees of species. There's another part of the evolutionary history of organisms that we frequently do not represent when we draw evolutionary trees, and that is we frequently don't draw all of the branches, even if we're showing species evolution. We may not show all of the species that exist for one particular tree. So if we go back to the primate evolution example, there are, in fact, other lineages that are known for different groups of primates. There are at least 2 described chimpanzee species. There are arguments all the time about how many gorilla and orangutan species there are. There are only, as far as we know, 1 modern human species lineage, but there are other things that have branched off, like the Neanderthal lineage and others in human evolution that represent distinct evolutionary lineages, but we don't always show those when we're trying to show primate evolution. We might just show a subset. We also frequently don't represent ancient extinct lineages when we show trees. So if we put that all together, there's a lot of complexity behind an evolutionary tree that is not represented in these simple branching diagrams. We will show you in the rest of the class that those simple branching diagrams are incredibly powerful for understanding the diversity of life, the mechanisms of evolution, the biology of organisms, and all sorts of other aspects of life. But the true evolutionary history is more complicated in most cases. So what I'm going to do in the last couple of minutes here is now walk you through the course in the context of a tree of life. And that's probably how you should think about the different parts of the course when we're talking about them, is in the context of a tree of life. So this is a very simplified view of the tree of life. There are 3 main branches of organisms in the tree that we will talk about in this class: the bacteria, the archaea, and the eukaryotes. We will discuss each of them throughout the course. We will discuss the structure of this tree with 3 branches throughout the course. Do you have a question? Or yeah. Again, that's a simplification. I'm not going to show you all the complexities behind that because we will talk about that when we talk about each of the individual groups in the tree. We are, in fact, going to expand extensively upon the eukaryotic branch of the tree in the tree of life. There are many distinct lineages that we will talk about in the eukaryotic branch in the tree. So in terms of the course, for the first week, we're going to talk about just trees and phylogeny and methods to infer phylogeny and how you can use them to interpret biology of organisms. So it's going to be concepts and methods to understand phylogeny and phylogenetic trees, largely because that's the rest of the course. We're going to assume you understand these things that are covered in the first week. In week 2, we're going to talk about the bacteria and archaea. I'm going to cover those topics. So 2 of the main lineages in the tree of life we will talk about in week 2. In week 3, I will cover what are sometimes called the protists, also called the microbial eukaryotes. These are all of the eukaryotes that are not plants, animals, or fungi. So all the rest of the diversity of eukaryotes we will cover in week 3. And then there will be midterm number 1, which will cover the material from those first 3 weeks. Then Professor Keene is going to spend 2 weeks on plant diversity and evolution. I will then spend a week on fungal diversity and evolution. And then there will be midterm number 2, covering those 3 weeks. And then the last 3 weeks of the course will be on animal diversity and evolution. And then the final exam will cover that material predominantly, as well as material from the rest of the course. So that's the big picture for the course. And what I want to do in just the last minute here, I'm not going to go through the examples that are in the slides, but I'm just going to tell you about it, and you can think about them, because we'll cover them in the next class, is that what we really want you to get out of this is diverse ways to make use of an understanding of the evolutionary history of organisms to interpret the biology and the biodiversity of life on this planet, and to understand the history of life on the planet. So it is, phylogenetic trees in and of themselves are not the point here. The point here is that they represent the history of life on this planet, and that they help us interpret and understand the current life on this planet. And that's what we will cover in the next few lectures.